The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, this is episode 70 of the eLife podcast. I'm Chris Smith from The Naked Scientist. This month, the drug that recreates key aspects of schizophrenia, enabling us to understand the condition in much more detail. The bitoscope, why scientists won't have to offer up their arms to hungry mosquitoes in future. And healing without scarring, researchers have found a key actor in the skin repair process. Before that, though, infertility is a major problem in developed countries worldwide. Just between 2017 and 2018 in America, fertility fell by 2%. This means that increasingly couples are resorting to assisted conception methods, like IVF, to help them to conceive. This usually involves collecting eggs from the woman and sperm from the man, fertilising one with the other, allowing the resulting embryos to develop for a short period in the dish, and then choosing the best-looking embryos to put back into the uterus to hopefully trigger a pregnancy. But this is a very subjective technique, and the success rates are still relatively low, at around 30%. And even among couples who do succeed, they usually need to go through several heart-rending and exhausting rounds of this process. So Hadi Shafii from Harvard wondered whether an AI system could help.
One of the most important factors is the ability to select the highest quality embryo. And the current methods to do that are either manual, highly subjective, expensive, and time-consuming. So we wanted to tackle this problem by developing a machine learning-based, fully automated approach to help embryologists to pick the best quality embryo.
Normally, an embryologist would look down a microscope at a selection of developing embryos, and they would say, I reckon that one looks like the best prospect, followed by that one. And those are the ones they would cherry pick to put back into the woman, aren't they? You're saying, can we use computers to help us do it even better?
You got it right. Actually, we performed a very simple but very important experiment before this publication that we had at eLife. We recruited several embryologists with different backgrounds, from 2 years to 8 years of experience, and asked them by providing them hundreds of images of embryos with known outcome, basically ask them to perform two simple clinical decisions that they normally do in their practice. And after they made their decision, we just rotated the images and asked them again to perform the same process on the same images. And there was huge variation between the decisions that they made.
So in other words, looking at the same embryo twice, they didn't make the same decision all the time, every time, showing there's enormous variation when they do it by eye.
That is correct. So one application of using AI is to literally help them to at least reduce the subjectivity of the process. So basically how the system works is that, we provide thousands of embryo images with its known outcome, and then the machine intuitively kind of figure out what kind of features it has to emphasize in order to make the right decision.
If you do a trial and compare its performance against what an embryologist could achieve, how much better is the AI system?
Very, very good question. So in fact, we did that study by recruiting 15 different embryologists from 5 different fertility centers across the country. And we provided images of embryos with really high quality. When you look at them morphologically, they look amazing. And we asked them to make their decision based on its implantation outcome. And we asked the same question from the machine. And we found that the machine actually did a better job statistically compared to the embryologist, 75% versus 60-something% on average.
That's quite a big difference. Do you actually know what it's looking for?
At this stage, it is literally a black box. We literally don't know what kind of features actually the machine was looking at.
Does the machine make any surprising decisions? Does it ever disagree with you where it chooses an embryo and you'd think, well, in a million years, I'd never put that in?
Very good question. Yes, in some cases, it does surprise, We literally saw embryos that actually, if you show it to an embryologist, the embryologist wouldn't transfer it, but the machine picked that, and then it ended up with a successful pregnancy. In some of the cases, it actually does surprise you in an interesting way rather than in a negative way.
So if this were applied to your average IVF clinic, where rates are currently running at about a 30% success rate, what sort of success rate would this return, all other things being equal?
In order to get a very confident answer, you literally need to perform a prospective clinical trial where you have a randomized study. But when you look at the retrospective data we have done, our machine showed that, we're doing better even compared to MGH Fertility Center, which is literally among top 10% fertility center across the country.
Well, it sounds very encouraging, doesn't it? Hadi Shafi'i there. People with schizophrenia experience a range of symptoms, one being difficulty weighing up different pieces of information in order to make an informed choice. Now, UCL's Sean Kavanagh has used both a computer model and tests on monkeys to show that low doses of the drug ketamine damps down the activity of NMDA receptors in the synapses that connect nerve cells in the brain to produce the same decision-making deficit that you see in schizophrenic patients.
The way that we kind of approached this to try to relate the problems at the synapse with the behavioral symptoms was that we built a computerized neural network, basically a computer simulation made up of nerve cells. It was made up of synapses, and these synapses had the NMDA receptors that we were interested in. We then used this neural network to test our ideas. So we said, what happens with this neural network if we interfere with the NMDA receptors? So this gave us specific predictions about how decision-making would change and how neural activity would change. And then we could subsequently test these in an experiment.
You used a computer to come up with some testable ideas, basically, that you could then go to a real brain and ask, if we do this, does this happen as the computer suggests?
Yeah, exactly. That's what we did.
And so what did you do to actually test it?
The research subjects that we used for our experiment were monkeys. And the way in which we tested it was by using the drug ketamine, because ketamine is known to block the NMDA receptors, and ketamine is also an excellent experimental model of schizophrenia. As we know, when ketamine is given to healthy human volunteers, it temporarily reproduces many of the symptoms of schizophrenia, such as the hallucinations, the delusions, and the cognitive symptoms.
What decisions were you asking the monkeys to make?
The types of decisions that we were interested in were those which involved combining multiple pieces of information. For instance, when we're deciding where to go on holiday, we have to combine lots of different attributes to make that decision. We might be thinking, for instance, of how excited we are about a possible destination, how good the weather will be, what the cost might be, and probably, unfortunately, at the moment, whether we'd have to quarantine when we return. So you have to combine all of these different features. Obviously, we didn't get the monkeys to decide where to go on holiday, but what we did get them to decide was on a sequence of images. They were showing some different bars. They had to add up the heights of all these different bars, combine lots of pieces of information, and decide whether a series of bars on the left or right of a computer screen were higher.
And your intervention is that you present exactly the same task, but with or without the presence of ketamine. So you can see what role the NMDA receptor that's going to be hit by the ketamine is playing in helping them to resolve various stimuli to make a decision.
Exactly. So what we were trying to achieve was to study how the monkeys make their decisions normally, and then through using the ketamine, because we know it simulates the symptoms of schizophrenia and it blocks NMDA receptors, that's basically giving us a window or an opportunity to what we think might be going on in the brains of people with schizophrenia.
Before you tell us what happened to the monkeys when you did this, if you ask a person who does and a person who doesn't have schizophrenia to do these sorts of tasks, what different outcomes do you get between the two groups?
So we know from previous studies they have impairments in decision making. They struggle with combining different pieces of information, they make their decisions off only a small proportion of the information they're actually shown. So if we come back to the example of deciding about a holiday, we had how excited you were, we had the weather, and we had the quarantine rules. If a person with schizophrenia was making that decision, they may only use the first attribute that they consider. They wouldn't consider any of the other attributes.
And the monkeys given the ketamine, did they show impairments in doing this simple decision-making task based on bringing information together in this way?
Yeah, they did. So it's important to remember that we used a very small dose of ketamine. So the monkeys were still very engaged. They still were performing the task. They're still enjoying the task. But they just became slightly less accurate in their decision-making. So when they were not administered with ketamine, they were getting it right maybe 85% of the time. When they were administered with ketamine, their accuracy went down to maybe 70%. But it wasn't just they were getting worse at the decisions. It was they got specifically worse in a way that we had predicted with our computer simulation.
Do you know why that is? And similarly, why a person with schizophrenia would struggle to integrate all these different bits of information together? Is it that they just can't hold all the information in working memory at once and therefore make valid comparisons? Or is there something else going on to explain why their decision-making is falling in this way?
We think that the NMDA receptor, which is a part of the synapse, is important for communicating between different nerve cells. And once this communication breaks down, the neural circuits, they're not able to combine information as well.
Does this point, Sean, to any ways in which we might present information in a way that is more meaningful for people who have this sort of problem?
It's important to stress again that we didn't test any people with schizophrenia in this study, although that's something we will be doing in future. But you are correct to say that for using these kind of sophisticated behavioral tests, we've worked out ways in which they may struggle to combine information. So it's also correct that we could also use these behavioral techniques to try to change the way in which these people were able to combine information. So that would be one approach. The other thing that is important to remember is that now we've identified something which we think may be going wrong in the neural circuits. The next step can be to work out how we can fix this. So we could think that if we had a therapy or some sort of drug with which could restore the activity of these NMDA receptors, then possibly this may be able to improve the symptoms of patients.
Let's hope so. Sean Kavanagh there. You're listening to the eLife podcast. I'm Chris Smith. Still to come, making skin heal without scarring, and how good are we at open access? There are thousands of species of mosquitoes, all with their own patterns of feeding behaviour, things that they are repelled by or attracted to, and diseases that they can carry. Studying them consistently and realistically is therefore a big challenge. But now Felix Holl from the Institut Pasteur in Paris has come up with a gadget that he calls the Bitoscope. This is an apparatus that's home to a horde of hungry mosquitoes and a patch of synthetic skin carrying a blood-like fluid. There's a camera below the skin patch hooked up to a computer that can document how the insects behave.
We now just use basically a thing that's most easily thought of as synthetic skin. So basically just a fake human mimic. To us, it just looks like a piece of plastic. But we treat the piece of plastic in such a way that mosquitoes find it attractive enough to find it, land it, and bite it. We, for instance, heat the substrate close to body temperature, which is a very strong guiding cue for mosquitoes. We also can spray in CO2, which is normally in our exhaled breath, and that activates the mosquitoes to start searching for a host. So we can guide the mosquitoes towards this little device, and there they land on a membrane. And the mosquitoes actually need to pierce through a surface in order to be able to feed. They insert their mouth parts, and here they find an artificial meal that is transparent. It has basically the essential qualities of blood that make the mosquito think it is like blood, and they actually engorge as if they would be drinking blood.
Where's the camera then? Is that underneath that transparent membrane so that you're basically seeing the mosquito from underneath as it lands and then tries to feed?
Yes, that's exactly right. So the mosquitoes are just contained in the cage, and in the floor of that cage, we have the bite substrate. We can just image through the bite substrate while they're feeding on the substrate.
And that data, that's all going into a computer. Does this mean that you can basically use image analysis on it so that rather than you painstakingly picking apart what the mosquito is doing, timing how long it does each step, you can get a computer to do that? So you can do this potentially at enormous scale because you could study loads of mosquitoes without actually having to do anything yourself.
Yeah, so we developed a lot of algorithms, a lot of custom software that picks out the mosquitoes in those images, where it was, how long it was walking on the substrate, when it starts biting the substrate. And from those images, we can also obtain things like how much does their abdomen swell. So of course, their belly swells while they're drinking. And from how much the belly swells, we can actually calculate whether or not it's drinking and whether it gets fully engorged or not. You can track where exactly all the legs are going, where the mouth parts are going, where the head is. And from that, we can basically calculate the relative positions of the body parts.
Do you think the mosquitoes are actually fooled by this system, though? Do they actually really regard this as skin, and are they behaving in a way that you think is exactly the same as they would were it your arm in there for real?
So I do think that indeed they're fooled. They binge on this artificial meal, they drink a lot of it, they swell 3 times their volume. So in that sense, they're clearly fooled. At the moment, we don't really know what the relative attraction of our substrate is compared to a human arm. I mean, that would be an interesting follow-up experiment to do at some point, where basically we present a real arm to the mosquitoes and we present our substrate, and we look at whether they prefer the arm over our substrate or maybe the other way around.
Now you've got this working, have you actually begun to gather data from it yet that are highlighting things that we previously, because of the constraints of the way we've historically tried to study mosquito feeding behaviour, had overlooked?
Yeah, so there's one example in the study that Anopheles colutzii, so that is a mosquito species that is important malaria vector in Africa. People had known for a long time that it is repelled by the mosquito repellent DEET. However, the mode of repellency was not entirely understood. And in this study, we actually show that the mosquitoes sense it with their legs. We could coat a small portion of the bite substrate with this insect repellent. And when they land just outside of the coated area, we see that they just walk around and have their normal behavior. Whereas when they land in the coated area, they immediately take off right after their first touch. So from this, we learned that actually the mosquitoes are likely tasting this repellent with their legs, and they're using that information to leave. And I think in the future, there's many different things that we can do with this setup. For instance, one thing that I'm very excited about is to compare the behavior of mosquitoes that are infected with a pathogen, for instance, dengue virus or malaria parasites, versus the behavior of mosquitoes that are not infected with anything. In the literature, there are certain reasons to think that infected mosquitoes actually may behave differently. Maybe they're more avid or less avid to feed. Possibly the infected mosquitoes will take a smaller blood meal and therefore maybe actually will need to bite more often and therefore actually infect more people. These things are fairly difficult to measure because it's pretty difficult to do experiments on infected mosquitoes. And we think that with the bitoscope, we will actually have a really cool angle on that and see if they actually behave differently.
Felix Holl on the bitoscope that they've just pioneered in Paris. Quickly healing a wound is a key part of our skin's defence against bacteria and other microbes that can cause infections. The price we pay for that speed, though, is often a scar. But can we make skin regenerate without scarring, or even reproduce complex mini-organs in the skin like hair follicles that are also often lost to injury? It must be possible because developing fetuses can do it, and as Eva Higginbotham heard from Washington State University's Ryan Driscoll, he thinks he's found a gene that might be the key to the process.
So there's a lot of research that was done by Michael Longaker and his mentor, and that were doing these life-saving in utero surgeries. And what he found was when those babies were born, they didn't have a scar, even though he had done surgery in utero on those fetuses. And that was a remarkable discovery. So one of the things we wanted to do was identify not only a molecular factor, but also the cell types that are lost during skin maturation that would convey this ability of embryonic or neonatal skin to be able to regenerate. And the first step that we did was we started to do single-cell RNA sequencing. So single-cell RNA sequencing is a new technique that's been developed so that you can sequence as much as possible all of the genes that are expressed within a cell. And then from there, you can sequence thousands of cells within a tissue to identify cell populations. And using that technique on, say, comparing all of the genes within a cell of over 5 to 10,000 cells in young skin, and then comparing that same assay in old skin, you could find new cell types as well as the genes that are defining those cell types. And that's what we did.
I see. So you found new cell types that were involved in really effective scarless wound healing in young skin.
I would say we rediscovered them, and we verified that they were there and that those cell types and that cellular state disappears in older skin. And we defined that cellular state by at least one gene. There are other genes that we found as well, but the gene that we decided to choose was LEF1. And they're there in the young skin, and they're not there once the skin matures.
So what is LEF1 doing?
Right. So LEF1 is a transcription factor. A transcription factor is a protein that binds to DNA. And many transcription factors bind to DNA to control the regulation of genes to be expressed or sometimes to be inhibited. LEF1 has been identified to be able to modulate signals from the outside of the cell called Wnt that come from other cell types or through its own signaling. And it's a part of a signaling pathway that's important during development and for some reason is somewhat turned off as the skin ages.
So once you found LEF1, what did you do next?
So the cells, some of the cells are still there. They just look more aged, right? They don't have the same phenotype. The state, the molecular state as measured by single-cell RNA sequencing is not there. So we hypothesized that if we could turn on LEF1 in those older cells and keep it on, then it's possible that that transcription factor could sit on the DNA and be ready for a signal such as those that happen during wound healing. So we collaborated and found a mouse that allowed us to control the expression of that transcription factor through, transgenic technology. And it allowed us to express LEF1 in those older fibroblasts. And when we did that, we created all different kinds of wounds at different time points. And except for one time point, all regenerated with this enhanced ability that we hadn't seen before with the erector pili, so that those wound hair follicles can even stand up if the mouse got cold.
Now that we know the importance of this factor of LEF1, what can we do with that information now? Do you think one day we might have a cream that has LEF1 in it that we can rub on our skin if we get wounded?
Right. I don't think you could put LEF1 in a cream because transcription factors are notorious to be able to kind of put inside of a cell, right? So the idea would be, and what we're trying to do now is to understand what LEF1 is doing in those special cell types, right? What are those downstream targets that LEF1 is activating in response to a wound? And then I think we would actually want to try to understand this in humans.
Ryan Driscoll. So no LEF1 anti-scarring cream just yet, but some very valuable knowledge nevertheless. eLife, the journal behind this podcast, is a digital pioneer. Since its launch, the journal has published exclusively online and it's open access as source, which means anyone, anywhere can freely access the research papers. Of course, many journals don't operate this way and they instead place their content behind paywalls, depriving those who have no subscription. Funders in many countries, however, have introduced policies in the meantime that the work that they're supporting should be made freely available in an open access format. So how is the research community getting on? Let's hear from someone who's been trying to find out.
My name is Chen Kai Kao Huang, and I'm from Curtin University. We started with the project wanting to know how institutions and universities are performing in terms of making their research output freely accessible to people. Through that process, we're able to pick up several things, including effect of policies and several other findings that should have implications to how we proceed in going forward in terms of making knowledge more accessible.
What was the period that you were considering?
The result that we have includes data that goes back 50 years, and the most recent 20 years are reported in the article itself.
And how have things changed?
The last decade, there has been a lot of focus on making knowledge accessible. Lots of research has gone into research showing that making knowledge accessible to people is good for making more knowledge. And there's been a lot of initiatives in recent decades pushing this agenda forward. And we see that in the data. There has been very low level of open access, maybe going 20, 30 years back. But in recent years, there's been a lot of push for that to happen. And we do see the open access level going up exponentially in recent years.
What's been the main driver of that shift towards more open access?
There's a lot of open access initiatives people have been pushing, and researchers are working very hard in terms of advocating for open access, not only in terms of showing research in terms of advantages of open access, but also implementing policies at regional levels, at country levels as well, able to push the agenda forward.
So how did you do this study? What did you actually measure?
Our data framework measures the percentage of output for each university in which they make their research freely accessible. We do this for more than 1,000 universities globally. What we found was that there are different policies and different resources that are having an underlying effect on how universities are performing. In terms of Australian universities, the current status is that universities in Australia lags behind universities in Latin America in terms of making research accessible through journals, but also lags behind universities, for example, in the UK, in terms of making publications available through repositories. A lot of that is to do with the strength of the policy and the infrastructure that's provided to support making things open access.
There's an old saying, what gets measured gets done. And if you're saying that there are different performers in different geographies, say Australia versus UK, is that because there are policies that also include monitoring in one place that are not present or so rigorously enforced in another place, and as a result, less research is translating into the open access space in one particular territory than another?
Yes, that's correct. Our data shows that places where there's more likely to be monitoring and possibly sanctioning of making research open access, places where the strength of the policy is stronger, they tend to be more open access. However, this is just one particular string of the story, because we suspect that it's a lot of combinations of different things.
What's going on in Latin America then that means that there's such a high representation of research output from there which is going open access? Is it because it is facilitated and incentivized and made cheap for institutions there, whereas a first world institution like an Australian university or a UK institution, there's a financial deterrent? Do we know?
For Latin American universities, we think that a lot of the open access levels of the universities are being supported by a project like the Cielo project, which has been providing infrastructure to hosting open access journals, which has made the whole cost of open access much more affordable. We think that is why Latin American universities have such high levels in gold open access, providing open access through the journals. Of course, there's the issue of funding, pushing through open access in a short term, but there's also examples after a few years, there's saturation.
What do you mean by saturation?
So we do see the effects of the funding, but that effect seems to slow down as time goes by and if there's no extra funding.
What would be your recommendations then? If there was a sort of a list of things that the best performers could teach to the worst performers, what would they be?
Well, the first thing we observed that really worked well and is cost effective is the providing infrastructure like in the case of the Cielo project in Latin America. That seemed to have a much longer term effect. So that's the first point I would recommend, to have some kind of infrastructure support for universities. Secondly, we have seen cases, for example, in the UK, even though there have been extra funding provided around 2012 for open access, a lot of the push for open access in recent years has come through university library repositories. So that's possibly another route we might want to support universities in going forward.
Carl Huang. Well, that is unfortunately it for this month. You've been listening to the eLife podcast that's produced by The Naked Scientists. There are previous editions as well as the references and full text transcripts for these programmes, alongside details of how you can subscribe to this podcast on thenakedscientist.com/eLife. And incidentally, The Naked Scientists also publish a weekly science programme that covers the latest leading science news stories. You can find that at nakedscientist.com/podcast. I shall be back with another look inside eLife next month. Until then, from me, Chris Smith, thank you for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.